I've been listening to the other presenters and I've been having a great time. And I think this is a very good way to discuss and in a friendly way put all our questions out there. So thank you for organizing. And also thank you for considering the new technology transfer program for the donations. I really appreciate that. And there's many things coming, many projects. So I'm excited to work with the Bee Breeders Association. So just give me one sec. So let me know if you are able to see the screen.
We can see the screen. It has the speaker notes next to it though, Nuria. There you go, that's full screen.
Yep, excellent, thank you. If something happens, let me know and I'll be happy to rewind and repeat or whatever it's needed. Okay, so I'm gonna talk about grooming behavior. Grooming behavior is a trait that I have been enjoying a lot to work with, and I had the great pleasure to work with great researchers along my career. So I'm gonna start with a very brief introduction about grooming behavior. Just give me one sec, it's a little bit slow here. There it is. So when we talk about honeybees, we have to think of them as individuals. Each of these individuals have ways to defend themselves against diseases through a very sophisticated innate immunity that help them defend themselves against fungi, bacteria, viruses, and also parasites. But as a honeybee colony, they have also developed ways to defend themselves. And some of these defense mechanisms are behavioral defense mechanisms, and some of them work very well against parasites. And one of these parasites is Varroa Destructor. I don't think Varroa Destructor needs much introduction. We know as beekeepers that Varroa is a very damaging parasite. What can I say? It reduces honey yield, it weakens colony. If bees are not properly treated in the Northern Hemisphere, they will collapse and die within 1 or 2 years. It acts as vector for viruses such as the deformed-wing virus, Israeli acute paralysis virus, black wing cell virus, and we are getting more and more. So there's no doubt Varroa Destructor is the #1 problem for beekeeping, particularly in northern climates. And so we have to focus on different ways to try to control this ectoparasite. And bees have different social defense mechanisms. For example, hygienic behavior, in which bees detect or identify diseased or dead brood. They uncap the cell, remove them from the colony, and it's a way to reduce the transmission of diseases within the colony. They also use the antimicrobial properties of propolis. There are some cases recorded of an increased temperature in hives in the presence of fungi such as Ascosphaera apis, which is the agent that causes chopper. And they have other ways such as absconding. That means just leaving the hive that has the disease and starting all over again. And we also have grooming behavior. Grooming behavior can be divided into allo-grooming, in which bees use a tremble dance or invitation dance to request help from their nestmates to remove parasites or particles from their bodies. And then we have self-grooming behavior, in which bees use their own legs and mandibles to remove things from their bodies, including varroa mites. Some of these behaviors, we still don't know a lot of them, a lot about them, such as bee fever. Recent studies have shown that we need to investigate more and confirm what's going on. Other ones are not very good for beekeepers. I don't think any beekeeper would like to select for absconding behavior. But others show a lot of promise to be incorporated into breeding programs, including hygienic behavior and grooming behavior. And the reason is that they are very effective to control the diseases. And also they have a genetic component. That means they are heritable, so we can select for these behaviors. So for this presentation, I'm going to focus only on self-grooming behavior. And just as an interesting note, grooming has been studied in other animal models, such as mice and rats, and has been a way to study aberrant behaviors, such as anxiety. So there's a number of things that we know about self-grooming behavior. For example, that is, it was first studied in bees in Apis ceranae, that is the original host for Varroa. And then we saw that, or it was identified in honeybees as well. It's effective to restrain Varroa Destructor population growth and also other parasites such as Acarapis woodi, that is a tracheal mite. We also know that the intensity in which bees groom is important to control Varroa. That means that if the bees use their more than 2 legs with a lot of energy to remove the parasite from their bodies, they have a better chance to get rid of the mite compared to bees that groom lightly. That means using 1 or 2 legs with very slow movements. We also know that the honeybee genotypes vary in their ability to express grooming behavior. For example, Africanized bees and the Russian bees, or the Primorsky line, are better groomers than European bees. And there have been genes linked to grooming behavior, such as neurexin. And just like any trait that we want to incorporate into breeding programs, we have to be able to measure that trait, just like we measure milk or honey production or other traits of interest. But how can we measure self-grooming behavior? Well, for research purposes, there are individual assays. They consist on putting a honeybee inside a petri dish. The original assay uses a Varroa Destructor. You put a Varroa Destructor on top of the bee's thorax, and then you spend 3 to 6 minutes watching the bee and recording the time that the bee started to groom, how much time she spent grooming, if she groomed lightly or intensively, if she was successful at removing the mite, and we then can assess the mites for injuries. But this is time-consuming and is mostly done for research purposes. But there are other traits that have been associated to self-grooming behavior, including mite population growth, the proportion of mutilated or chewed mites, and the severity of those mutilations. So for mite population growth, what we do is record the mite levels in the spring, then 12 to 16 weeks after, in the fall, we subtract those numbers and we calculate how the population of Varroa grew within a season. That's mite population growth. We can do it with any technique, for example, alcohol wash or sticky papers. If we use sticky papers, then we can also look for chewed or mutilated mites and calculate the proportion of mites that show injuries, and we can also rank those injuries. These 3 traits can have other variables or components associated to them, for example, natural mite fall or mutilation due to allogrooming or just other things that happened in the colony. But the important thing is that all of them have been correlated to self-grooming behavior. And then, because we have found genes linked to self-grooming, there is a potential of using molecular markers to assist the selection. There's a lot of researchers and studies that have been done with grooming behavior, but if I had to choose just few to understand or to summarize what's going on in a practical way, I would choose these ones. This here, the authors found a correlation between the proportion of chewed or mutilated mites with self-grooming behavior in a lab assay. And then this one from 2012. It was done by Dr. Ernesto Guzman at the Honeybee Research Center and in collaboration with a university in Mexico. This paper is interesting because they found a link between the colony phenotype, or the behavior of the colony, and the behavior of the individuals. They found an association between mite levels, the proportion of mutilated mites, with the performance of grooming behavior by individual bees. And they also found that the bees that groom intensively had better chances at removing the mite, and there were more intense bees coming from presumably resistant colonies, which were Africanized bees, Russian bees, and a stock selected for low Varroa population growth. And they also found differences between genotypes. They found that the ones that I mentioned, the Africanized bees, Primorsky line, and low Varroa growth selected bees, were better at grooming and had lower mites compared to European non-selected stock. So that's good news. And then this study here is interesting because they found a link between a part of the chromosome with 27 genes and grooming behavior. And one of these genes was neurexin, which has also associated with self-grooming behavior in mice. And later, in other studies, they found, just give me one sec. It's a little bit frozen here. Okay, there it is. Then in this study, they found that bees that groom intensively had higher levels of expression of neurexin compared to light grooming bees or no groomers. So I'm gonna focus on two breeding programs that have incorporated self-grooming behavior or traits linked to self-grooming behavior, because I had the chance of working with them in one way or another. And well, one of these projects started in the 90s with Dr. Greg Hunt at Purdue University in Indiana. This project was, well, at the beginning, they incorporated a stock that was composed mostly by bees or colonies from the Midwest US. They also had Russian bees and bees selected for Varroa sensitive hygiene, or VSH. And this selection mainly consisted on selecting for the proportion and severity of mite mutilations, although throughout the years, they also incorporated other traits such as self-grooming behavior or mite population growth. So that's why the name of the Indiana mite biter stock. And this, in these photos, we can see Crispin Gibbons assessing mites under the microscope to observe mite mutilations and rank those mutilations. This stock has been evaluated twice, one in 2014 and the other one in 2018, and they found similar results. This stock was compared to Carniolan bees and Italian bees to assess how the breeding program was doing, and they found that the Indiana mite biter stock had better overwintering success. They also found that the Indiana mite biter stock had a higher, sorry, I'm just looking at the graph and I'm trying, I think I made a mistake, but the Indiana mite biter stock had a higher proportion of mutilated mites, lower levels of mites, and lower mite population growth. And they also found a correlation between the gene neurexin and the proportion of mutilated mites. So this is good news, and that means that the selection was working, and they had good results and practical results for the breeding program. And then in parallel, Dr. Ernesto Guzman at the University of Guelph was also investigating grooming behavior, and he was working on a selection program. In this case, the selection was bidirectional. That means that they were selecting for low Varroa growth and high Varroa growth. And one of the differences between the the two programs was that at U of G, the selection was mostly focused on mite population growth, although they also incorporated other traits such as mite mutilation, severity of the mutilations, and female mice reproducing in the cells. And they also evaluated other health parameters, including overwintering success. So this program started in 2018, and the preliminary results are available in an open access article that you can find. So if you Google some of these words, you can find it and download the articles if you want more details. This project started with Dr. Berna Emsen. It was led by Dr. Ernesto Guzman, and is currently, all the work is being done by Álvaro de la Mora. He's a PhD student, and he's doing a great work doing all these assessments. And briefly, as I mentioned, this project has the characteristic of having both selections for low varroa growth, which is what the beekeepers want, but also high varroa growth. And the reasons are for to understand better the mechanisms behind behavioral defense immunity, and also to confirm that the program or the project was going in the right direction. And just as to summarize this project, just give me 1 second, I'm gonna move this here. And this started in 2018 with Generation 0. They used more than 300 colonies to do the selection. What they did was record the mite levels in the spring, then again in the fall, calculate Varroa mite population growth, and select 6 low Varroa growth and 6 high Varroa growth colonies. The first population, or the populations that they used to start, included bees from different backgrounds, including Apis mellifera ligustica, carnica, and the buckfast strain, which is bred at the Honeybee Research Center. Then they overwintered these colonies, and from these, the colonies that survived, they chose 3 from the low Varroa growth and 3 from the high Varroa growth selection to graft and produce 150 queens from each genotype. They used colonies from beekeepers that participated from the Ontario Bee Breeders Association, and they divided the colony, balanced the population, the health, food resources, etc. And they introduced a queen from the LVG to 1/2 of the one division, and 1 HVG queen to the other division. And then they again did all the evaluations. They recorded mites in the spring, in the fall, calculated mite population growth, and then selected again 6 low Varroa growth and 6 high Varroa growth colonies that were overwintered. And then in 2020, they repeated the process all over again. There are different ways of controlling mating, including instrumental insemination, using an isolated area such as an island, that is what they use for the buckfast strain, or drone flooding. In this case, they used an open mating system, and what they did to control the paternal side was to not have any other colonies or any other source colonies in a radius of 5 kilometers. And what else can I tell you? They assessed other health parameters, including overwinter mortality, but also hemocyte counts that are related to innate immunity, deformed wing virus levels. And there are a couple of other parameters. Female mites reproducing in the cells, and also hygienic behavior. And in a summary, what they found in this first generation was that the low Varroa growth colony has an increase of only 1.7 Varroa mites, compared to the high Varroa growth that had 9.6. Also, the low Varroa growth colonies had lower levels of deformed wing virus compared to the high Varroa growth. And as you can see, the mortality was lower again in low Varroa growth compared to the high Varroa growth population. So this is a summary of what happened for the selection program. And if I could summarize the practical things or aspects that bee breeders can use, would be recording mite levels might be consuming, but it's worth it. And if you use sticky papers or alcohol wash, you can do two assessments in the spring and then in the fall, at least 16 weeks apart, to be able to calculate Varroa mite growth. And then select from the colonies that have the lowest mite population growth, and consider this trait as an additional trait from your selection program. You can do a pre-selection with traits that are of importance to you. For example, you can pre-select for hygienic behavior, defensive behavior, overwintering success, honey yield, and also consider low Varroa growth in your assessments. Other thing to consider is that to do this type of selection, you need a big population to start, so more than 100 colonies. As you can see at the Honeybee Research Center, they started with more than 300 colonies, but I guess 100 colonies might be a good place to start. Also, it's good to consider management practices. It's not the same to have colonies that are used for honey production than colonies that are used for nuke production or queen production. Any management that breaks the cycle of Varroa Destructor will impact our calculations on the mite population growth. And it might not reflect the genotype or the potential of the colony. It might be a reflection of our management practices. So it's just something to keep in mind. If you're assessing your stock, make sure all the colonies are managed in the same way. And also, it's good to consider this low Varroa growth selection as a long-term strategy. There's no rush. For this selection process, it was done very quickly. There was a lot of pressure to do the assessments and identify the colonies that they wanted to use. But it can be done over time, and also test your colonies and make sure that they are performing the way you want to perform. And also considering that if you're incorporating other behaviors, you have to keep them in mind as well for the assessments. And then, just as a side note, one of my roles in these studies was on the molecular aspect. So this is more of I don't know, for me, the fun part of these projects as well. But what we did is took advantage of having the low and the high Varroa growth, so the extreme genotypes. And this was to know more about the mechanisms behind this behavioral immune response. In other words, behind grooming behavior. So instead of using a Varroa mite to do the evaluations, we used flour. In this study that was done at the National University of Mexico, we used flour to differentiate grooming behavior between two genotypes, which were Africanized bees and European bees. And we noticed that flour was good to contrast both genotypes. So, and it was also, it's also easier to do it in a lab setting. And it's funny because Varroa mites is something that you don't want. You're always working to keep them under control. But when you need them to make assessments, they are not there. So there's a challenge. So just give me 1 second. I'm trying to run this video, but I don't know why it's a little bit slow. There it is. So this worker bee is grooming intensively. She's using more than 2 legs to remove the flour from her body. And, and she was using intense movements to get rid of the irritant, which in this case was flour. So this bee, after doing this process of grooming behavior, we froze her and we conducted some molecular analysis. And we found interesting things that are still under evaluation, and hopefully we'll have the publication soon. This study was done in collaboration with Purdue University with Dr. Brock Harper. And we saw that the most informative trait to differentiate the low Varroa growth and the high Varroa growth was time of first grooming, which confirms previous studies done on this behavior. We also identified potential markers and some proteins that have also been identified to be involved in hygienic behavior. So they are probably proteins that are used to identify the irritants or the varroa mites and trigger a motor action in the form of grooming behavior in the bees. We found some new insights on how this behavioral immune response works, and hopefully we'll have more detailed information to come. But the main aspect of these programs is to have tools available for beekeepers. When we select for a trait, that trait has to be easy to measure and also in a cheap way or practical way. So it appears that calculating for Varroa growth is a good way to quantify a trait linked to a defense mechanisms to control Varroa Destructor. The next step would be to contrast the Indiana mite biter stock against the low Varroa growth stock from Guelph and have more insights on how these mechanisms work. We also found interesting things about the viruses that are infecting or that were present in the central nervous system, that means the brain of the honeybees that were evaluated. And we know that we have to understand more about viruses and how that infection could affect immune behaviors, such as self-grooming behavior. And then the practical aspect of these studies are to push or to have more information to build a marker or a genomic-assisted selection. These type of selections have been very successful in agriculture, for example, or to select for cattle. They are good at identifying economically important markers to assist breeders in their selection. It's not a tool that will replace the behavioral assessments, but it can complement or enhance records. And another thing to consider is that it's difficult to select for a single trait or a single marker. Complex behaviors such as behavioral immune responses, they don't come in one gene, they come in many genes or in many chromosomes that are influencing these behaviors. So we have to be careful about our selection. But it's a good way to start, and we're having more information about these mechanisms. So we're hoping that at some point we can assist breeding programs with these tools. And so just another thing to consider about complex traits such as grooming behavior is that they have a genetic component. So they can be heritable, they can be passed generation through generations, we can select for them, but they are also influenced by environmental factors. In this study that was conducted at the University of Manitoba, Dr. Curry found that temperature and humidity were affecting self-grooming behavior. Then in another study that we did at the Honeybee Research Center, we found that sublethal doses of clothianidin were also decreasing the proportion of self-grooming bees. And following up with this investigation, because we saw that bees that were exposed to this neurotoxic insecticide not only had reduced self-grooming intensity, but there were pathways related to neural processes that were affected, and we were curious about to know how and why. So we follow up with another research, this time in collaboration with Memorial University of Newfoundland, and we found that clothianidin altered self-grooming behavior. We confirmed that, but we also found that the lipids that are in the brains of the honeybees were also affected. Lipids are very important for the function of the central nervous system. And we found lipids linked to energy, to the stability of nicotinic receptors. It's, well, a number of things that could be linked to alterations that translate in poor self-grooming behavior that can compromise the health of a colony. So this is in a nutshell what we have found, and our interest in self-grooming behavior. And I guess the next step is to keep looking at ways to facilitate the selection for these traits and assist breeding programs with tools that are economic and practical. And as a conclusion, this self-grooming behavior is a quantifiable trait, and we can do it through traits that are linked to it, such as low mite population growth or the proportion of mutilated mites. And honeybees are a great animal model to understand neural processes, and self-grooming behavior has given us a lot of information about behavioral immune responses in this great social and interesting insect. So I think this is a summary of what we have been doing, and I hope it was helpful. And I also, if you want to know more, the Honeybee Research Center produced a series of 3 videos that you can find in YouTube. So if you just Google LBG University of Guelph, you'll find those videos. And the first one is the theory on self-grooming behavior. The second one is Dr. Berna Emsen explaining how this selection was done. And the third one is Les Eccles explaining more of the practical aspect of breeding and incorporating LVG into breeding programs. So thank you for your attention. And well, here is the list of our collaborators, including the technology transfer program of the Ontario Beekeepers Association. So thank you. And if you have any questions, please let me know.
Thank you very much, Nuria. That was awesome.
Thank you.
Yeah. It's great to get all the papers and all the background information as well. Now I think we'll let Wendy and Emily Huckster take over for the questions if you're ready for those.
Yeah, for sure.
Okay, thank you.
Thank you.
I'm just trying to get my video to work again. But I have a question, Nuria. And it is from a beekeeper. And it is, what is your advice to the general beekeeping population on how we can all support bee breeding and improvement in BC stock? And I think I could add, I think I could add on to that by and say that what we're trying to do is lessen our dependence on imported stock and also lessen our dependence on bees that need a lot of Varroa mite treatment. And we're doing pretty good at that, but not 100%. So we would appreciate your input.
Okay, for sure. I'm making notes. I'm just taking notes to make sure I don't miss anything. But I'm going to start with the second part of the question that, or the interest, that is selecting or not having, or having bees that don't require a lot of chemical treatment, if I understood correctly.
Yeah.
One of the integrated pest management strategies, one of the main components is genetics. So genetics is the first tool that beekeepers have to protect their stock against Varroa mites and other diseases. So incorporating traits that can help with this is very important, and it's doable. There are mainly 2 traits that have been identified to be heritable. That means that they can pass through generations, that you can quantify that trait and select for them, which are hygienic behavior and grooming behavior. So there's a lot of potential on that. And it's not that genetics will solve all the problems, but it will give more room to decide when and how to treat, and depend less and less on synthetic acaricides. Then the other part is about sustainability and breeding. So that is a huge question. And I was hoping the opposite. I was hoping that breeders could tell me what they need and how TTP can assist them. But I could say that in my experience, or what I have seen, is first is organizing. Second, educating, that I think you're doing a great job by doing this, just having these conversations and discussions and just ideas on how we can improve. And I guess through education and growing and collaborating, there can be a big improvement. What do I mean by this? Some beekeepers might not be big enough to have a big population to select from, but the whole province have enough stock to select from. So we have to take advantage of what we have and use that, and also project for short and long-term goals. Where do we want to go? What do we have? And what are the steps that we need to get to that point? But I'm hoping we can keep discussing the strategies, and I think there's more to come on that topic.
It's nice that you're working with us.
Well, thank you. I'm very excited for all the things that are coming, so thank you.
Here's another question from a beekeeper. Are you aware of Dr. Kara Wagner's research on brood odors and developing a more beekeeper-friendly hygienic test?
I am not.
That's okay.
But I'll be happy to
we can find out.
that. Yeah, I'll look for the name in the chat. Perhaps I've read about that, but it's not on the.
That's okay. We can look. We can find out for sure. What do you think about environmental aspects having an impact on the grooming behavior? Are there any environmental aspects?
If there are environmental stressors that can affect grooming behavior, yes. So grooming behavior is interesting because they have calculated the heritability of the behavior. That means the genetic component. So all that we are, how we look, as organisms, it's mainly due to our genes and to the environment.
Right.
So the genetic component has been calculated, but has been very variable. And they have calculated a high genetic component of grooming behavior and very low. But what we know is that we can select, and that we can see that selection or that improvement in our stock, and as fast as 1 or 2 generations. But environmental stressors can affect self-grooming behavior. And we can see it, as I mentioned, the study from Dr. Rob Curry at the University of Manitoba. They found that temperature and humidity were affecting self-grooming behavior in lab assays, if I remember correctly. And then again, in lab assays, we saw an effect of clothianidin on self-grooming. And in another study, we also saw an effect of neurotoxic insecticides in other behavioral immune responses, such as hygienic behavior. So stressors can affect the traits, and there's something that we need to consider, for sure. I hope that answered the question.
It does.
Just let me know.
Thank you.
Sure.
Someone is asking if you can see the chat feed that's on right now. Can you see it?
I can see it.
Oh, Carrie Clark is asking, is there a commercial stock with low Varroa growth that you know of, and good honey production and good temperament?
I have a feeling that the next presentation will be better to answer that question. So my background is more on the research aspect. So I had the great fortune to collaborating with Dr. Greg Hunt and now with Brooke Harper with the Indiana mite biter stock. And one of them aims is to have that stock available for beekeepers. And I believe through the Heartland Association, they are doing that work of sharing the genes.
It seemed to be, yeah.
And then from the UFG part, the UFG side, the idea of this program is to have a protocol that beekeepers can use to do their own selection.
They're real beekeepers and bee breeders.
Yeah, exactly. So the tools and the information are available.
Yeah.
I mentioned these 3 videos that are good as a reminder and just to have tips or tricks on how to do it. And the article is open access if someone is curious to see what happened and how they did it. But yep. And also those tools will be incorporated for their ORBS projects or programs. So that's the idea.
That's a great idea. It's so exciting how you're working to use science to help real world beekeepers and bee breeders. It's very nice.
Yeah, it's always interesting to see the whys, but we also have to look at the problems at the industry and see how we can advance and help for sure.
Right. I'm not sure how many more questions there are.
Well, I see one here is the drones groom. I would say yes, but I think their proportion in the colony is very slow to have an impact on the control of varroa mites. So I don't think they've ever looked at them to assess grooming behavior. I think in many studies, drones are not accounted for something. But I was preparing a presentation for another conference, and I wanted to focus on drones and selection for the paternal line, because we tend to ignore them, but they are very important for breeding.
Do you know how long it takes for their grooming behavior to make the mites fall off and fall down? Is it like right away, or does it like take a little while?
It takes a little bit. So we give for research purposes, we give ourselves a limit, because you can spend a lot of time
Waiting.
watching a bit. So the standard is 3 to 5 minutes. But I don't recall what we found, to be honest. But yeah, some bees are very quick at reacting. I work with flower, they take seconds to react and remove the flower. For mites, it can take a while. And also mites are tricky. They move, they hide, they go between the sternites. So the bees have to invest a lot of energy to get rid of them. But there's a video that one of my colleagues took, and when she presents and the mite finally takes the mite off the body, and you can see the mite on the floor of the petri dish, everybody claps and get excited.
Going down, yeah.
So there's a couple more questions I saw here. Has anyone compared the effectiveness of the mite biter versus the VSH type bees?
Compare the effectiveness for controlling mite population growth? Let me think about that question. That's an interesting question. I think it would be hard to differentiate them within a colony, because the colony would have both traits and they would perform those behaviors. So which is better? I don't know, and I can't recall of a study that has been doing that, but I can go back and check if they have. I know we can have compared different aspects of the behaviors. Again, for example, how like the genetic component of the behaviors. But yeah, that's I'll take that as a homework.
No, fair
Nuria?
enough. Another question I saw here that I thought was really interesting by Carrie Clark again is it necessary to control for the age of the bee when you're looking when you're comparing their grooming trait?
Yeah, for example, for research purposes, we need to control as many variables as we can to have the effect or know the effect that we're looking for. So for example, for the clothianidin study, all the bees were 7 days old, for sure, the same age. Then for the other project, what we did was use brood nest bees. So we knew they were not old enough to be foragers or going out. We used bees that were in the frame, but we didn't know exactly their age. And we used a bigger sample size for those studies.
So do you have an exact, I might have missed this when I was reading the other people's questions and stuff, but do you have an exact test to test for my biting bees that beekeepers can do?
Okay, for sure. Yeah. So the idea with the University of Guelph program is to have a very practical way for beekeepers to use.
Good.
So having a beekeeper putting a bee in a petri dish and looking at her to see if she grooms or not, that wouldn't be realistic. But there are traits that have been linked to this behavior, and one of them is mite population growth. So you have to record the mite levels in the spring, in the fall, subtract that number and see how much they grew within a season. So that's a trait that has been linked to self-grooming behavior and that beekeepers can use to do this selection.
And
okay.
I have a quick question about that, Nuria. So if you do that, do you treat for mites prior to that first Varroa assessment,
Yes.
or do you leave those colonies as is as they come through the winter?
The best way is not to treat them for the mite population growth and then treat them right after your last record. So for example, in this case, all the bees, the bees were, the mite levels were recorded in the spring, 16 weeks later, then right after they were treated with Apivar. Now, I know it's hard to do that realistically, because if your mite levels are going crazy, you don't want to compromise your stock. So just make sure that the bees that you're assessing are having exactly the same treatment. So if you're dividing colonies, take that into consideration. Divide all the colonies, or don't do that management, or treat them equally, because you don't want any other variable to interfere with your measurements. Because you wouldn't be looking at the genes of your colony, you would be looking at your management. I don't know. I hope that makes sense.
It does. Where does the grooming usually take place? Like right at the entrance when the mites are like coming in on the foragers, or somewhere else around the brood nest?
It mostly takes place inside the hive, like in the brood chamber. So as you remember, mites emerge with the bees. So that's when they would start grooming or ask for someone to groom them.
Right
so
away.
Mostly brood nest, but it depends, and it's a hard question because mites are everywhere. So whoever has the mite and has that reaction, she will do it. And also mites are going from one bee to another. They don't remain in the same bee. So
that's true. That's true.
Yeah, and for studies, I guess we want bees between 7 and 14 days old. That's when most grooming happens, but that doesn't mean that it couldn't happen in foragers or other ages.
Well, we're really looking forward to working with you.
Well, same here. Thank you for the invitation, and this has been great. Thank you.
Yeah.
There are a couple more questions. I think
Oh,
one that I really liked is Doug Gordon asked, is the intensity of grooming associated with any other trait, for example, aggression or the amount of brood produced in a hive?
So there has been, but I think this needs to be confirmed, and they are working right now on that. But one of the traits that they were assessing for the at the University of Guelph was defensive behavior. So it seems like the genotypes that are better at grooming, they are also a little bit more defensive. So that might be linked. And yes, when you're selecting for a complex trait, it would be very difficult to just have that one. You would most likely be selecting for other ones that are inherited with the trait that you want. Yeah.
I think that sort of addresses your question as well, Ian Lowe, but if that isn't true, feel free to anyone can just jump in and ask their own question as well, just if we're missing questions there.
definitely.
Yeah,
I haven't looked down at the bottom.
and I guess just to add, I'm just scrolling down to see if we missed anything, but just
I see that Jerry Rosima has raised his hand for a question.
Yes.
Yeah.
Okay, I think I've got my video on here. We see this all the time, and this may sound like a really dumb question, but every time we get a talk like this, people talk about selecting for survival as a trait, winter survival. I just want to ask, how do you select against survival? Because it strikes me that the dead ones are dead and we can't graft from them. So is it just something that you always put on as a bullet, or is there actually something to that bullet that we don't understand? That's my question.
yeah, for sure. That's a great question. And there's of course, when for example, if you're looking for a colony to graft, well, they have to be alive. There's no other question. But when we're doing research, we're working with populations. So when we're comparing, for example, the low Varroa growth and the high Varroa growth, we're assessing those traits. And we're looking at 150 colonies and checking the percentage that survived and the percentage that didn't. So it's more of a evaluation of the stock. And yes, of course, you cannot breed from the death. Yeah. So I hope that makes sense. It's not that we are selecting for success or unsuccess, is more of an evaluation of the trait.
Another great question here is, are different Varroa populations more or less susceptible to the grooming?
That's a good question, and we don't know.
Okay.
We don't know, but just like a heads up, and if someone wants to participate in this study, please do. So we're gonna launch soon this citizen science study to calculate Varroa economic thresholds in British Columbia. There's going to be two aspects. One, the on-site, I'll be doing the assessments, but for the other part, we'll need the participation of beekeepers and to give us the information. But part of this project is we're going to be collecting samples of Varroa mites, and we're going to be looking at their genotype and at the viral levels and see different correlations between mites and bees. So that's a, yeah, I think it would be hard to answer your question about if some Varroa mites are more susceptible to self-grooming behavior, but in a bigger study, I'm sure there could be something. Now, these traits are difficult because they are complex. And a honeybee colony, there's a lot of things going on, including VSH behavior. And also, just to add, I think to add to Fred's question, is just a complement to that. If you select for low Varroa growth, you can also select for other traits. So you for sure can select for Varroa sensitive hygiene, for hygienic behavior, and for low Varroa growth.
And are there any side effects to grooming, negative side effects to grooming that have been found so far, such as spring buildup, their inability for spring buildup or honey production?
Not that we know of, except for defensive behavior.
She said their nastiness possibly.
If
A
you're
little
okay with-
defensiveness is always good.
But it might be a might. So I'll keep an eye on what Alvaro, the PhD student that is working with the colonies, finds. I'll let you know how it goes.
And
Yeah.
how can we find out how to collect samples for you for that?
Oh, well, thank you. Yeah. So I'm going to be releasing all this information soon. So in the B scene for next month, this month, I'm lost in which day is today. But in the next B scene, there's going to be information on how to do that.
Okay.
And our website is very close to being ready. So I'll let you know when that happens. And all the instructions and information will be there.
Perfect.
Thank you.
And another question I saw here that was really interesting is, what's the heritability of the algal grooming traits?
For the grooming trait, I think it has been done for self-grooming, and it's very variable. It went from, in some early studies, it went from 0 for allo-grooming to 0.4 for self-grooming.
Okay.
So there's a range there. So I think it would depend on exactly what trait linked to self-grooming they use to do the assessment.
Okay.
But so the closest one to what we are doing, the trait that we're assessing would be around 0.36. And just as a reference, hygienic behavior has been calculated between 0.18 to 0.65. And I remember a study of 0.39. So around the 0.34, 5, they are good. Like they are medium heritability, but we can work with that.
Okay. So fairly usable. I see Ian Lowe has his hand up. Go ahead, Ian.
Sorry, I was a bit distracted. I've got a little guy next to me. But anyways, I missed a little bit of the question, but I'm curious, like, how do you distinguish between, I know just before you're mentioning that temperature and humidity have an effect on grooming behavior. And in terms of locally adapted or even the colony traits, I don't know, it's not necessarily a genetic thing that I'm looking at. Is there such a thing as a social learned kind of personality kind of thing that's affecting or can be measured in here too, or that's possibly corrupting the data? I don't know. I'm just kind of wondering what's out there.
For sure. Let me see if I can answer. But for the temperature and humidity study, if I remember correctly, this was done at the University of Manitoba, and they use lab assays to assess self-grooming behavior. So it was not at a colony level. Now, if there is a non-genetic trait in the colony that might be affecting self-grooming, I don't know the answer, and I don't recall any study related to that. And again, sometimes we don't get into that because these are complex. Like there's a lot of things happening in one colony. So
yeah. So, and the other part of the question is, you mentioned temperature and humidity are important. Like, is there a sweet spot of temperature? Like, do you have a range? Or even for humidity, is there a range of, I don't know, like 20 to 50% humidity? Or do you know what those numbers are?
To be honest, I don't remember the numbers in the study, and I don't know if those would apply at a colony level. I hope that makes sense. But I have a list of things to look for, so I can add that to the list.
Yeah, because like even parking your bees in the shade in the summer, like how is that going to help, kind of thing? Like, I'm kind of curious about that. So anyways, thank you.
Yeah. I don't have the answer, to be honest. Yeah, but I guess if minimizing stress just in general, I think I wouldn't, I think that would be helpful.
And another question I see here that we missed is there a standardized flower test that a beekeeper could do, or a method for doing that?
There's a standard protocol that we use for research purposes. If you're curious on seeing it yourself, I can for sure share the articles. The last one that I shared is open access, and you can see what we did. Now, for practical things and for selecting yourselves, I wouldn't suggest the grooming assay with flower. It's time consuming. I had to look at more than 1,000 bees to to do the assessment. So I would suggest the mite population growth, and it also gives you more information about the health status of your colonies and when and how to treat. So it's not a bad practice to do that anyway and see what Varroa is doing.
Hopefully everyone is doing that anyways.
Yeah. I wonder too if maybe an assessment that's easier, and somebody has kind of recommended this or has suggested this, that the hives where like the bees are kind of chewing back the wood, or you put in a pollen patty and they eat every last bit of the paper always, if those ones are also good groomers. I mean, not necessarily, I realize that leg movement might not be associated with mandible movement, but
yeah, I guess we would need a lot of hives to assess that and to see if there's a link that is significant to those behaviors. And then again, like all the variables around those things are hard to measure.
Nuria, do you think you'll be able to set up a research apiary through your job?
So we thought about that, and the idea of the TTP is to cover the whole province. So having educational apiary around this area will only cover the needs of a small population. So we will be looking for partners to do workshops and to have some knowledge transfer and tech transfer in different parts of the province. So we're hoping that beekeepers will be willing to host workshops and work with beekeepers in their area. So we're thinking more in a moving around.
Right. Covering the whole province.
Exactly.
Well, thank you very much, Nuria. This has been a super good presentation. I think we've all gotten a lot of takeaways from it. And I want to point out to people as well that this series is given freely by our speakers. But if you would really like to support the TTP, go ahead and you can donate through the BCB breeders and those funds. Anything donating from this series will go toward the TTP and supporting their efforts for breeding queens. And so I, donate if you like what you see. And then I should also introduce next week's talk, and it will be by Dorothy Morgan, who's the president of the Kentucky Queen Breeders Association. And they have a really good citizen science project, more or less, going. They're looking at feral bees, and how they collect feral bees is very interesting. Then how they actually do their breeding, and then how they hand out or give access to beekeepers with the bees that have these traits. And as well as Dorothy has lots of great videos about bees grooming right at the entrance. She has a video of that as well, and many others probably. I'm looking forward to her talk next week to kind of give us the opposite side from research, a citizen and a queen breeder's aspect to the same trait. So I'm looking forward to that, and I hope you all are. And with that, I think we'll sign off. We've done our hour plus here, but it's been great. Thank you, Nuria.
Thank you for joining us for this presentation. BCB Breeders is producing a series of recorded sessions to share knowledge within the scope of breeding honeybee queens. For more information, check out our website at bcb.org. bcbbreeders.ca. To support ongoing research to be shared with beekeepers across Canada and the world, please click the donate link on our website at bcbbreeders.ca. Thank you.